Good morning. My name is Rohan Uttamsingh, and my colleagues are Justin Meyer and Zeyu Zhong. We have all just finished our freshman year here at the university, and we are investigating the interactions between microgravity, actin structure, and infection.
So our research question is, does the actin deformation caused by microgravity increase the susceptibility of cells to infection by pathogens that rely on actin deformation for infection?
So, as background, we know from past experiments that microgravity can lead to the deformation of actin, which is a cytoskeletal protein that is important to cell structure and movement. However, in these past experiments, the cells have only had seconds of microgravity exposure. So we would like to investigate this further through our experiment with weeks aboard the ISS. Additionally, many pathogens manipulate the actin structure of their host cells in order to facilitate infection across the host. With the large number of pathogens that manipulate actin structure, these researchers wonder what the impact of an already weakened actin structure caused by microgravity exposure could be on the infectivity of such a pathogen.
So for our experiment design, before we send our FME up to the ISS, we're going to observe the actin structures of the cells by first staining them, which will allow us to see the actin under a microscope, and then analyzing them using a procedure called fractal box count analysis, which will just allow us to quantitatively measure the actin structure of the cells. Then when we send them up, we will send one FME up to the ISS and keep one down on Earth just to increase the validity of our results.
So the samples will consist of a type 2 FME, which is a tube that contains a clamp separating two solutions. So in one of the solutions, we will place 3 milliliters of essential medium with KCO2 cells. The essential medium provides the nutrients for the cells to survive, and the KCO2 cells are cancer cells of the epithelium type, and they're in the colon. And in the other solution, we have 0.5 milliliters of pure mycin. So this is a growth inhibitor that will basically freeze or preserve the structure of the cells and allow them to remain the same. So this is useful because before the cells come back from space, the astronauts can unclamp and mix the solutions up so that the cells and their actin structures will be preserved as they return to Earth and they won't revert to the original state. And the same procedure will be done for the sample on Earth to keep the experiment valid, so that the only thing that is changing is microgravity.
So once the cells come back down from the ISS, we will again analyze both the control and the ISS sample using fractal box count analysis and record any differences between the two. Then after we record that data, we will subject both samples to Listeria, which is a pathogen that manipulates actin and spreads between cells by using the actin to form sort of a comet tail that allows it to propel itself into different cells and infect them. While this is happening, we'll measure the infection rates under a microscope using something called time-lapse video microscopy, which will just basically take a time lapse over 20 hours of the infection. And then after, we'll analyze that quantitatively and qualitatively, focusing mostly on rate of infection, differences between rate of infection, and differences between methods of infection of the Listeria.
And for the applications, so in the future, as space travel becomes more common and accessible, it would be beneficial to understand the impact of microgravity on human cells and the actin structure and how that could lead to increased susceptibility to infection. And also, this is one of the first times that the effect of microgravity on actin structure and that potential increase in infectability of pathogens is tested at the same time.
Thank you all so much for your time. We hope you're as excited about our experiment as we are, and we wish the best of luck to all our fellow participants.